Thank you all for being here. Welcome to I think this is the final Emerging Research Showcase of 2022, focused on commercializing academic research, which is a fairly unique niche, also known as bubblegum and duct tape, and we'll explain that in a little bit. It's my very great pleasure to introduce Steve Lommel. Steve is the Associate Dean for Research for the College of Ag and Life Sciences to start us off. Steve?
Thank you, Deborah, and good morning, everybody. I'm excited about today. I'm just here to give you a little bit of introduction from the college. I see some faces I don't know, but and maybe you don't know about this program, but this is something Deborah's been running for a while, these research innovation showcases. They tend to often have a theme. This one is a little bit more of a potpourri, a collection of our earlier base technology that's probably been published on, and there's been intellectual property filed on it, so we can talk about it. But again, this is more of a little bit of a collection, and hopefully I'm not spoiling her words, but I think that's the reason for the term bubblegum and duct tape. But we'll hope you enjoy the presentations today. I think we have 5 different programs that are going to be talking about their new inventions and their new programs. But what I'd like to say from the college level is this really, this demonstration today that we have and that we're showing really comes to the core of what we're trying to do here in the college with interdisciplinary projects, science, but also developing technologies and getting them out. It sort of fits with our university theme called Think and Do. And I'll stop there and thank you all again for coming and turn it over to Adrian Percy, the director of this facility and the Plant Sciences Initiative. Thank you.
Thanks, Steve. Thanks, Deborah. Especially thanks to Deborah for all your hard work to get us to this point. I'm really, really excited about today, and it's great to see so many people in the Plant Sciences building. Welcome. We hope that if you've not been here before, hopefully you can get some exposure to the facilities during the day. I think there is always options for tours at some point if people want to do that, but you are all very welcome. We are very proud of this facility, although yesterday I was at the Alexandra AgTech facility and they informed us that we're only the 3rd nicest AgTech building in the Triangle. So it just shows that we're pretty, we have an embarrassment of riches here in the Triangle, if we can say that. So I wanted to tell you a little bit about the initiative before we get started. But one thing I did want to say up front is the reason we wanted to put on this showcase in this particular way was to really mobilize, our community. We have an incredible community and ecosystem here in the RTP area in North Carolina and even nationwide when it comes to AgTech. But it really will take a village to achieve some of the things that we need to achieve. We have some great scientists here, as Steve said, great engineers, great data scientists working together on early stage technologies, but we need help getting it from where they are today to something that can make an impact in a farmer's field here in North Carolina or across the US. And that's where you guys come in. We know we have investors here, we have technology savants and folks who have done this before many times in some cases, and many entrepreneurs in the room. So please, if you see a project that is of particular interest to you that you feel you could help out on or you have some advice to give, then I can tell you all of the faculty here, the students who are presenting are all ears and they're really hoping for that. So to tell you a little bit about the Plant Sciences Initiative, for those of you who don't know, this is a interdisciplinary effort at NC State. This building was only opened in April of this year. We moved in around 25 scientific programs over the summer. We're more or less up and running, although we still have a few parts of the facility to bring online. But really the intent of the Plant Sciences Initiative is to go after some of these really intransigent, difficult problems that we see either in agriculture or in our environment. And we believe that we can only get at those through interdisciplinary science and through bringing together folks from across a wide breadth of backgrounds and perspectives. Can I do this or? There we go. Thanks, Anthony. So we have a pretty big vision here. And again, to achieve that vision, it's going to take a lot of folks from a lot of different parts of NC State, but also different organizations, whether they're agribusiness, whether they're investors, whether they're growers, whether they're different types of stakeholders that all have a voice and all have a role to play in terms of, helping us overcome some of these grand challenges. We work in a team-based concept, which means that this building, I get pushback when I say this, is kind of an Airbnb for research. We bring groups in, they work together, and hopefully they can go after some of these big issues, but we're always looking to collaborate, to form partnerships with entities either across campus or exterior to the university. This is just a kind of snapshot of where we are now. We have a number of the colleges across NC State who are represented with different types of expertise that are working either in this building or under different programs under the umbrella of the Plant Sciences Initiative. As you can see, the red dots here are the College of Ag and Life Sciences. We were born out of CALS, but we really see ourselves as part of the interdisciplinary movement that we see at NC State at the moment. And you can see also, we have a lot of engineering contributions. In fact, 25% of our faculty involved in programs here are from the School of Engineering or the College of Engineering, but we also have other colleges, the Colleges of Natural Resources, College of Sciences, College of Textiles, all represented in various interdisciplinary projects that we have going on. We did a quick survey recently about, the types and the breadth of expertise that we have available to us, and this is kind of a word cloud just to give you a flavor of that. It's not exclusive, but you can see there is a lot of ag tech, biotech, data science type applications, and that really gives us the freedom and the opportunity to go after, as I said, some of these really big challenges, and trying to bring these groups of faculty together around big ideas to go after big grant opportunities, and to do that in collaboration with others is really what we're about. We've decided to create 5 platforms, 3 of which are research-based. All of them are faculty-led. So you can see on the left here, plant improvement. That includes a whole raft of different things around making crops more resilient, for instance, to climate change, bringing new varieties to growers across the nation. Technologies like gene editing are kind of housed under that particular platform. We have one around resilient agricultural systems, think sustainability. Chris Rebo-Corton, who put a lot of effort into this today but couldn't be with us, unfortunately, is leading that platform. He's working on things like cover crop management and other topics, but also working with others who are working on things such as soil health, carbon sequestration, these types of projects. Kranos Williams, who's actually a professor out of engineering, is working on our data-driven plant sciences, and data science really underpins everything that we're doing here. And we have suites of bioinformaticians, computer biologists, who are working with our biologists, with our ag economists, with our engineers to bring some of these technologies forward. And then we have 2 non-research platforms. One is around education and workforce development. And here what we're trying to do is give opportunities to students of all ages, K through 12, undergraduate, graduate students, to actually get experience in interdisciplinary working and hopefully build career-ready individuals to go either in industry or into further careers in academia. And then last but not least, we have an extension outreach platform as well under Rachel Van. What Rachel is doing is ensuring that we are listening to our stakeholders in the field, that we're understanding what are the agricultural issues that we're trying to solve for, that we're developing technologies which are meaningful to growers and not something that is unusable or not user-friendly. And then we, of course, want to work with industry and with others to scale that up into commercial opportunities, and that, of course, is one of the reasons that we're here today. One of the missions that we intend to have is to connect, and this is a great example of that, bringing together so many different folks from across so many different parts of our ecosystem. And we want to continue that. We want to bring together folks from our agricultural community here in North Carolina, which is very diverse, very varied. It includes commodity groups, it includes growers, it includes groups like Farm Bureau, Golden Leaf Foundation, and others. Of course, we're working across NC State, all the colleges, all the departments, and also interdisciplinary efforts that are going on at the university. We work heavily with the global ag industry. We have collaborations in place with companies like BASF, Syngenta, Bayer, and others. But we're also very open to working with companies of all sizes and all stages of growth. And last but not least, we're looking to work with both national and university academic partners as well. And again, this is exemplified, for instance, with our Science and Technology Center that we have looking at phosphorus sustainability called STEPS, which is working with a number of different universities across the US. And we're not stopping there. We're actually still working a little bit on this building, even though it looks pretty much finished from the outside. We are actually just yesterday started construction of a partners suite. This suite has ideation spaces, innovation spaces that will welcome industry partners who have either collaborations ongoing or want to have collaborations with our faculty here. And it's basically a touchdown space so that technology scouts, collaboration researchers can have space in this building and work alongside faculty. And hopefully that proximity will lead to new, better collaborations over time. We're also working on entrepreneurism. As you will see today, there is a whole bunch of different projects ongoing, and we really hope that we can help facilitate that, provide support. We have a huge number of resources and programs here at the university to support entrepreneurism. NC State is one of the country's leading institutions when it comes to commercialization of science, and we want to support in our world here at the Plant Sciences Initiative as best we can. We're building out an ag incubator right now, but we're also grateful to those of you who have been voluntold or volunteered to actually help out with advice and mentorship around some of the ideas that we're working on. And so this is where we are so far. As I said, we've only just opened. We've had a really successful time at raising competitive grant funding over the last couple of years, even before this building was opened. We've now got 25 programs, 4 USDA programs included, working under the umbrella of the PSI. We have a number of very interesting core facilities in this building. We can do genome sequencing, we can do confocal microscopy, we can do metabolomics, proteomics, and other types of technologies as a service to our faculty, but also to external entities. And we've even got 2 certificate programs. So we're running a certificate in ag data science and one in regulatory science as well. So there's a lot of things going on. Again, very, very happy to have you guys here. If you want to learn more, Deborah, who is here, is really helping us connect with industry. Dinah will be talking later about some of the advancement opportunities, but we have social media and we have newsletters and that kind of thing. So if you want to learn more about events that are upcoming, then please tune into that. I think now back to you, Deborah, right? With that all said. Thank you very much.
So, we got us. Can we have my slide set, please? That's me. Thank you. So, if you're reading the agenda, you have already realized that I am not Chris Rehberg-Horton. So, Chris has this. Honestly, this event was Chris's vision. We're really disappointed he wasn't able to join us. He's sad as well. But what I really want you to know is that Chris is the one that started with the bubblegum and duct tape idea. And so it's my job to introduce that vision to you and just remind you that as faculty, we're rewarded. We're rewarded for teaching, for extension, for research, for scholarship, and maybe that one. And oh, by the way, for commercialization. But you probably know commercialization is not necessarily straightforward, and it can be awfully challenging to resource. So Chris and I, along with the Office of Research Commercialization, who's here from ORC today? Raise your hands. Tim? Deborah, thank you. And I think Brian Eller will be here later and maybe a couple more. So we went back to the ORC team, and then Chris and I and the entire team went back to the faculty and said, what have you got? Is it a widget? Did you maybe publish it? And then, it was put together with bubblegum and duct tape, sometimes literally. And is it on a shelf somewhere? Or is it a technology that's not quite ready for prime time? You've done it, you've got all the stats, you've got all the data, but you haven't turned it into the app. So what we did was we asked our faculty to dust those things off and take them out today. You'll see a bunch of them in the expo, the Innovation Expo, up on the first floor, the higher part of the first floor. Is there a word for that? And outside, we've got the RASP unit, the mobile cow shelter, which is actually really cool. And so I hope during the break at 11, you'll take a chance, take an opportunity to go and see some of those expos. And of course, the 5 faculty members that are presenting today. Now, it's a really tough segue from bubblegum and duct tape to technology readiness levels, but I'm going to do my best. And 2 disclaimers right up front. One is that we're focusing here on what Kaltaran Chauhan in the ORC calls deep tech. So this is academic technology that is being spun out. And the second disclaimer is a personal disclaimer. I don't know any of this, okay? Engineers are trained in technology readiness levels. The rest of us are not so much. But so I've done a little bit of my own research with, again, Kaltaran's guidance. And I looked and looked and looked for the slides that I'm going to present to you today, and I couldn't find them, so I made my own. What are technology readiness levels? And this is stolen from USDA NIFA, Crop Research Technology Readiness Levels. They come out of the NASA system. There are 9 levels. Number 1 is the earliest stage of development. Number 9 is the highest stage of development, fully implemented. NIFA is actually moving to requiring you to fill out a form on what technology readiness level your technology is before you apply for a few of the programs. And interestingly, they process through 4 of what they call activities: preliminary technology, experimental testing, pre-commercial assessment, and commercial deployment. This one's mine. Okay. So these are the 9 technology readiness levels. 1 and 2 are things that we can generally fund through federal or state grant funding, maybe through a foundation grant. 3 and 4 and 5 is when we start thinking about university spinouts or technology licensing, which can happen anytime after TRL 3. My role, which I probably should have told you up front, is introducing our faculty to industry scientists with a goal of developing a research collaboration. So I'm working way at the very early part of the TRLs, 2 to 3 to 4, mostly maybe 2 to 3. ORC, and correct me if I'm wrong here, but ORC is working more in 3 to 5. So we're obviously, as a university, we're way early in the technology development stage. But everybody talks about the Valley of Death. So, okay, where's the Valley of Death? Well, the Valley of Death is way, way past where most of us are. It's after a company is spun out, and if you can make it through the Valley of Death, in theory, you've got successes of business. So I've overlaid 1, 2, and 3 under research. Again, these are not definite, borders, but 1 to 3 is research, 3 to 5 is development, and we don't even come close to the Valley of Death as academics. Again, the caveat here is deep tech. Nobody talks about the Gully of Death. The Gully of Death is where you've got a technology. Again, we're focusing on faculty here. The faculty member has a technology. It's a cool technology. They've done it in Arabidopsis. Arabidopsis isn't a crop, so they have to cross the Gully of Death into a real crop, in order to make the next step. Trino's going to tell you about one of those. Yeah, he's chuckling. You knew I was going to pick on you, Trino. There may be many gullies of death that a faculty member faces before they can even create a licensable technology. It's not as wide, it's not as deep as the Valley of Death, but you still have to throw resources into it in order to cross. and get to the next step in commercialization. And I'm using commercialization more generally. It's commercialization or other non-academic application. So here's where we come to the ask. My ask, all of our ask of you today is, first, to provide insights on what gully of death are each of our faculty presenters facing. Any of the Innovation Expo presenters, what gully of death are they facing? Is it the right gully of death? Maybe it's not the right gully. Maybe they need to be choosing a different approach. And what I'm hoping you'll see in yourselves today is a technology that you are interested in finding a way to help resource to get to the next step through commercialization or other non-academic application. With that, it's my pleasure to hand it back to Adrian Percy.
Thanks, Deborah. All right, with all that preparation, we're going to get down to it. But first, I'm going to invite our sharks to make their way over there. So just to introduce, and I'm going to let them introduce themselves, but just to say who they are. So we have Spencer Morn, co-founder and managing director of Finisterre Ventures. We have Heather O'Keefe from Novozymes. And Novozymes are great partners of ours and actually have space in this building. So we're really happy to have Heather here. And then Mark Brooks, who is with FMC Ventures. Their job is to stimulate, firestart a little bit of discussion. I will be turning to you guys if you have thoughts and questions, but maybe we'll start with one of the sharks before we turn to the audience. And just to reassure them, we're not expecting you to put your hands in your pockets and come up with $1 million investment here. It's just to give advice. That's what we really need at this point. So I think with that, we're going to call on Mike Kudenov. I think you're up first, Mike, and I know. Oh, sorry. Yeah, come on up, Mike. But let me, Mark. Why don't you just introduce yourself quickly and let us know who you are.
Hello. Thank you, Adrian. Thank you, Deborah. Thank you, Steve, and everybody for the hospitality and the invitation here. So I'm Mark Brooks from FMC Ventures. FMC is one of the big ag input companies. We make insecticides, herbicides, fungicides, all kinds of crop protection products. The belief that we have is that agriculture is part of the solution story around climate change, and that the future of crop protection is probably using less volume of chemicals and more precision agriculture applications, more biologicals, more things like that. And so what the venture capital team is looking for innovations at the early stage, like seed, Series A or Series B stage companies globally that can be the game changers of ag tech. And so we're investing in these companies as a way to learn, as a way to add value to their innovations and go to market together or co-develop things together, and also achieve some kind of a venture capital return profile. So excited to be here. I'm going in completely cold to these conversations that we're fixing to have, so I have no idea what I'm about to hear, but I got the pen ready for the check writing.
That's great. Well, that's good to hear. Over to Spencer.
Great. Spencer Morn, and is this on? Is that working?
Yeah.
Okay, good. I can never hear properly. As Adrian said, co-founder of Finisterre Ventures, which is a Silicon Valley-based venture capital firm. We focus on early stage companies, and I would say, we focus mostly on the food value chain, but really it's a problem that we see across industrial segments in that, when you're producing commodities, you have a lot of constraints around you, not just in the cost curve, but also just the fundamental sort of purpose of trying to produce more with less always. And that could be less on, impact on the environment, but it might simply be, a water constraint, a land constraint, all these sort of simple things, all the way through the value chain. So that's sort of what we spend a lot of our time doing. And then myself, just so everyone knows my biases, I'm trained as a geneticist. My thesis was in Arabidopsis, which got a lovely reference last night. I'm sorry, just before. I'm still on Israel time. I flew in just right now. I would say, yeah, it is tough to get to that jump into crops, so I empathize with the comment even more. Maybe we should try to make Arabidopsis a crop. It might be easier for everyone, but that's me, so.
Heather.
Wonderful. Thank you so much for having me. I'm really looking forward to this. So I work for a company called Novozymes. We are a world leader in biological solutions. We are the largest manufacturer of enzymes and microbes in the world. Our focus is really on how do we harness the power of nature and bring these sustainable solutions to life, whether it's improving agriculture yields, low temperature washing at home, to food, sustainability, and all the above. So really looking forward to this session.
Well, thank you to our sharks, and now we're going to turn over to our faculty. I know you know how this works, but just so everyone else does, we've got 5 minutes for a presentation, then 5 minutes for questions. Deborah will keep the timing, and you don't want to mess with Deborah. It doesn't end well. So, so Mike, over to you. Thanks.
Okay.
All right, I'll just dig right in. So my name is Mike Kudenov. I'm here, a professor at electrical and computer engineering here at NC State. And today I'll talk about integrating decision support into graded horticultural production systems. And really this is part of the Sweet Apps program, which was funded by NC State's Grip for PSI. So just to kind of first review the situation, stakeholders really have very heavy investments, upwards of $1 million, in existing packing facilities and optical sorters. Data is typically saved as a daily aggregate, so they don't necessarily save data in such a way that they can look at historical trends, and if they do, they usually keep it in their head, like as a mental model. And if they do have optical sorters for grading, automated grading, they may not necessarily be accessible to them. It might be proprietary. And so stakeholders have tons of investment in their current workflows. This would be like hand harvesting sweet potatoes for Scott Farms. This is also Scott Farms. They have these big production facilities for picking out, bad sweet potatoes, and they have this 12-lane sorter here for, kicking off or fulfilling orders for the day. Whereas this other facility, this is Nash Produce, has more of like a manual system. They don't necessarily have optical sorters. And so really there's a wide range of technological integration that exists both in the machinery and the optical equipment. And so really what we're trying to pitch here is an idea for a startup where the vision is to really be a world leader at reducing food waste within these existing agricultural production systems. And the mission is to accelerate the transition to more sustainable agricultural practices for providing easily integratable sensors, often in case open source, inexpensive sensors, with potentially proprietary data management and decision support tools that can really be layered onto these existing processes and really minimize the impact to stakeholders' workflow and facilitate adoption of the technologies. Resources, we have tons of algorithms that we've developed as part of the Sweet Apps program for field, bin, and belt grading using cell phones and fixed cameras. We also have data ingress, processing, decision modeling, visualization, and storage methods, and agricultural modeling and domain expertise. So this is like a flood of stuff that we can do. We can do grading of sweet potatoes in the field with cell phones. We can do grading of the so-called top bin of these trucks. So we come in, we get actual quantitative assessments on a truck scale after harvest. We can look at now grading on these big conveyor belts. Again, even if they do manual sorting, we can still plop the sensor in and get data. And we can convert that data into quantitative metrics. So this would be like an actual grade. And this is fresh off the press. We just made this visualization yesterday. And then also looking at putting them on automated harvesters, so getting things like yield maps, something that would be more typical in, say, corn or soybean. And then we have the data part. So we have tons of data. So we have methods now to ingress that data, produce visualizations, integrate these data into machine learning pipelines, managing the data, storing it, and then also assessing it in terms of things that are difficult to quantify, like shape. We can get length, width, we can get weight, but shape also relates to yield quality. Field mapping, for instance, relating all these data back to the field, soil types, environmental parameters, and then also yield modeling so that we can actually take these metrics and predict, when's the best time to harvest, how do we deploy our resources efficiently. In terms of activities, it's really using these emerging methods to provide decision augmentation or decision support, again, that integrates into stakeholder workflows in such a way that's least impactful and maximizes profitability. We also want to expand to other crops beyond sweet potato. Sweet potato is important for North Carolina, but this is something that we want to do. And then also classify surface defects. In terms of impact, then, short term, we want to formalize these cyber-physical system modules, the sensors and algorithms, so that we can, again, drop them into these existing processes with a minimal investment to stakeholders so that we can maximize the adoption of these technologies. And then really expand, at least within the sweet potato production system, the number of stakeholders that we have so we can diversify the preferences that we are accommodating. Medium term, we want to expand to other cropping systems. So many other cropping systems are graded: tomatoes, apples, peanuts, pickles, of course, cucumbers. And then long term, we want these tools that we're developing to be relied upon by the stakeholders so that they want more. We have RGB cameras, we have emerging sensors, VOC sensors, and you're not going to be able to pitch a VOC sensor, like a volatile sensor, to a stakeholder if they don't really have that existing background that these technologies are beneficial. So this is just showing there are many other different crops that are graded: peanuts, tomatoes. All these systems look very similar to sweet potato. Assumptions: we assume that stakeholders will be receptive, that we're fulfilling stakeholder needs, and that these technologies are extensible to other production systems beyond sweet potatoes. And we have integrated stakeholder engagement as part of the project. So we are fairly confident about the first two bullets, but really the third bullet, in terms of our ask, we're really looking to validate some of these assumptions on a broader scale across multiple crop systems. And then we're also looking for potential funding so that we can really maximize the impact of our vision and our mission and the work that we've done thus far. So with that, I'll take any questions. Thank you.
Do you have any from over here first?
Sure.
Mike?
I always have questions, but
Go.
okay. So what's the thing that I missed, I guess, in that presentation was the current state of the art and which component you're really focused on that's a step change.
Mm-hmm. Yeah, so I think like the current state of the art, for instance, I think the current state of the art in many of these production systems is a mental model. So it's the stakeholder looking at the crop, looking at the bins, assessing, producer X usually produces this kind or this grade of sweet potato. I have an order today for this grade. I will use this grower. And that's I think is the state of the art, at least within the sweet potato production system. And so what we're trying to do, again, is put that data layer on top so that we can better inform those decisions. Another example is the trucks. The truck will come in, they'll get a little ladder, they'll look at the crop, looks like a medium. So the whole truck gets assigned that classification.
Okay, I'll just have one comment and then I'll pass it. But yeah, I'd say there's a question in my mind that could be true for sweet potatoes. I don't think that's true for the industry. You can go to cherry sorting, for instance, in Washington, and they're like super sophisticated. I was amazed at the different software and data platforms that some of the big groups are using there. That happens in different countries, certainly for apples already. Even timber in the Antipodes, they do a lot of this, which was surprising to me. Point being, I think there's a question to say, are you going to build something either, licensing consulting based for sweet potato because the differentiator is that people aren't looking at that market, or are you trying to do something that is broadly applicable, which I think there's probably some more thinking to do around what is lacking from all of those. And it is just a word of caution. It's usually better to have one thing rather than everything. A lot of people think the differentiation is easier if we do the whole thing. And that tends to be pretty hard to do.
Yeah, that's a good point. One of the areas we've been thinking also about focusing on is underground root crops. So, onions, the sweet beets, the beets, another thing that we've been recommended as well. So
cool.
One thing I didn't hear, or maybe I just missed it, is how exactly is this helping solve the problem that you framed up, which was around food waste and reducing food waste? I think I missed that piece of the puzzle. And how big of a problem is that in sweet potato?
Yeah, so I suppose a good example would be, let's say they made the assumption based on their mental model that grower A produces the grade they need to fulfill today's order. They'll start dumping that grade onto the packing line, and by the time it gets to the end, they say, oh, we're not getting what we need. So now they have all this material on the packing line. It's already been washed. And that means once you wash it, it's done for in 4 weeks. If you don't wash it, you can store it for a year. So that material is done. If they aren't getting what they need, they have to switch fields. Everything on the line gets wasted. So making those decisions at the earliest possible time, right as they're dumping that bin or even before they dump that bin, would save, we estimate upwards 5 to 10% of what they're trying to produce.
Just a couple comments. So how are you helping the consumer in the sense of you're adding additional complexity to this? From what I understand for your solution, you're giving information, but that's not necessarily insights, right? So there's a step change for the consumer to adopt. And for that additional complexity of the crop that you're trying to identify and sort, what's that next stage? So you're giving a piece of the puzzle. So as you look to bring it to the consumer, think about the entire customer process, right? Because I think you have one ingredient, and in terms of adoption, that's going to be a huge block for adoption because info's great, right? Data's great, but what do we do with it, right? So I think you need to be able to tell that story and give a very clear vision on what are those next steps.
Okay, yeah, thank you.
Okay, sorry. Thank you, Mike.
Okay, thank you.
I told you not to mess with Deborah. I did. Okay, so next is Edgar, Lobaton. Edgar, please come on up. ForaBot.
Well, thank you very much. My name is Edgar Lobaton. I'm an associate professor in the Department of Electrical and Computer Engineering at NC State. So I'm going to be talking to you about ForaBot. This is an automatic foraminifera isolation and imaging system that we've been working with our students. So let me tell you a little bit about what foraminifera is and what our motivations are. So this is actually something that paleoceanographers use quite a bit, and I actually didn't know what a paleoceanographer was until I actually worked with one. So these are the forensic people in the ocean, and what they do is that they get samples from the bottom of the ocean, right? Basically some of this mud. Different layers means different ages, of course, too. They can do some carbon testing associated with that. And what they need to do is they need to get some specific samples of these microscopic fossils that you see right over there. So these are very small, right, in the order of like 500 nanometers to 50, micrometers to 50. And the idea is that right now, the standard is that they need to do this manually in terms of separating the different types of species, and different species tell them something about biodiversity. They also can do all sorts of chemical testing. And things that are useful for that is, for instance, to identify whether a particular site could have the potential for oil. That would be one thing. But climate change, monitoring, all those things is also very important for them as well, too. Right. So here's an example of what they need to do. They need to go through those samples, and they right now, again, do it manually. They sit in front of the microscope, and they need to separate it one by one. Of course, this is very tedious and very difficult to get very consistent type of measurements. So they, of course, tell the undergraduates to do it. But the undergraduates, of course, there's only so much that they can do, and we want them to be more excited about the things that they're actually doing right now. So what we're trying to do is that we're trying to create a robotic system that can actually automate that process, and that's in a very reliable way. I'll show you some examples in a second, but basically this table at the bottom, don't look at the numbers too much. Different species that we classify, and highlighted diagonal means that we're actually doing a pretty decent job. So we get very decent performances for this type of work. So the system kind of looks like what you have in there. We actually have a prototype outside, so you can actually get to see it if you want to see it in operation. And the general idea is that we're creating, doing something kind of similar to what Mike was talking about. We're not doing at that same scale with like conveyor belts, but we're trying to sort each one of the samples and then be able to separate them and organize them at a much finer scale. And of course, this is small, right? So The way how we're manipulating things at this scale right now is basically using suction, but we also have a prototype that we've been working on using microfluidics. So of course, this could be used for a lot of other things like cell counting and all these kind of things, right? So we have the manipulation portion of it and the sorting portion of it. That's the mechanical portion. I'm just kind of going to show some of the components. I'm not going to have time to go over all the different pieces that we have in here, but there's basically microcontrollers controlling the suction so we can actually raise a single forum, be able to identify, do some sort of imaging in there. We do different type of depth imaging, some new imaging that we actually came up with ourselves as well too. And then based on all that information, we're able to classify them, but also be able to get some information about their morphology. So I just wanted to briefly show one of the images that we're getting to. This is basically the kind of imaging that you get with a microscope that is gonna be like, at the very least, tens of thousands of dollars worth it. But the prototype that we're doing, and right now we're making this open source, is under $2,000 in parts. We're trying to basically expand it and make it available to people so that we can actually use it for other type of purposes as well. And then, of course, like the business portion of it, would be once we start having the data, also maybe being able to manufacture some of these and be able to make it accessible to other people as well. So besides the real type of data portion of it, and for people that are working on kind of the data portion of it, they know that getting data sometimes is one of the big challenges. How do we get all this data to be able to train these big machine learning type of algorithms, the deep learning, and so on and so forth? We've been doing a lot of data augmentation by creating synthetic data sets as well too. So that's part of the thing that we're doing. And we've been complementing basically our real data with the synthetic data so we can get to the point that where we can actually use basically our deep learning algorithms. So things, again, we're looking for is not just recognition, but we're trying to extract some information about the morphology in here too. So if it was cellular cells, for example, as well too, we could also do that as well too, extract a structure from each one of the samples. And just wanted to say that besides this type of work, of course, a lot of the technology that we do also transfers to other type of crops for phenotyping. I just wanted to kind of show that. But in general, I guess the opportunities that we're looking at is that we have in our group a lot of expertise for visual sensing, 3D phenotyping at the microscopic scale as well too, and we're interested in transferring these to other use cases as well. We do know, as somebody mentioned, that each specific use case requires a lot of fine tuning. There's a specific challenges for each one of them, and that's what we kind of like specialize on. And I think I'll stop right there. Thanks. Any questions?
Just a quick question. What was the really hard thing that you sort of cracked here to be able to open this up? And how's that positioned, I guess, from an intellectual property point of view?
So there's components in terms of the manipulation portion of it. There's people that have been trying to do this for like 20, 30 years, and now the technology is becoming much more reliable and available to be able to do that. So the manipulation portion of it, there's some challenges that we actually like overcome quite a bit in there too. The other portion of it is also kind of what would be more of the machine learning and the AI portion of it, to be able to develop like more algorithms that are kind of getting together with the machine to be able to give us reliable measurements of what species we have. So just to give you a little bit of I don't want to go in a lot of details, but or I can, I guess, if necessary. But what happens is that when humans are doing a lot of this annotation, there is a lot of uncertainty, right? So a person, for example, you'll get tired of doing the annotations, are going to start making mistakes, right? So the algorithms that we're working on are trying to quantify human uncertainty and making sure that the machine also captures that uncertainty, so we can very robustly, I guess, say that this is actually the type of species that we have. Yeah.
There you go. Look. Any other questions from the sharks, or shall I go to the Heather? You look like you have something.
Yeah. In terms of application, is it just academic in terms of application? So if you're looking to bring this to market, right? Who's your target market? It seems like it's very focused right now. It could have broader possibilities, but that's very unclear, right? So as you're thinking of your go-to market, there's some basic things along those lines. And is your key differentiator the fact of not only you have this machine learning, but it's the type of lens and the cost to bring it to market, or it's unclear. So I think that's something to hone in on. Who are you trying to target? What exactly is your differentiator, and what's the value proposition, right? Especially if this technology is becoming more available what makes yours different? Yeah.
Great. Thanks.
So I'll riff a little bit on what Heather just said. Nice presentation, by the way. I don't think I got the problem that you're solving at a big scale. I feel the way I soaked it in was kind of more of an academic application, but maybe I missed that. So if I missed it, then think about that and how you frame it,
absolutely.
the big problem you're solving.
Yes, absolutely. And I think the motivation from our end came mostly from that academic side of things, where there's basically hundreds of hours of people doing these annotations that are kind of, in some sense, they could be avoided, right? So we're trying to speed up that process, and this would allow us to give better information about like climate change and things of that sort.
Put that in there at the beginning then.
Yeah, thanks.
Any questions from the floor? Please, Ian.
What's the throughput of the system? And secondly, have you tried it on biological samples, seeds, embryos?
Can you just repeat
Yes. Yeah, so the question is what's the throughput of the system and what other samples have we used it on, cells or things like that. So this particular setup with the suctions has been mostly for Foraminifera, which is a microscopic fossils. They're very specific, the type of suction and all of that, so it's customized for that. The type of microfluidics that we've been testing, that has been tested before in other type of for example, cells as well too, blood cells and things of that sort. And that works better for like the smaller size fractions that we are going to be working on. And in terms of throughput right now, so the suction mechanism right now, it takes about like minute or so to be able to do the image of a single sample because we're mostly optimizing on trying to get really good images right now. We haven't optimized in speed. We should be able to get it to like a few seconds for each sample, but we're imaging one at a time, right? We're not doing a conveyor belt type of application.
So I think we have one more question. I saw a hand over here. Ron, please.
I was just curious, is the image analysis part of this applicable to, say, water sampling to identify Cryptosporidium and a range of different pathogens?
So is the image analysis applicable to water sampling?
Yes, absolutely. I think somebody asked this a while back, I guess, as well too. Yeah, but we could certainly do that as well too. Yeah.
Thank you, Edgar.
Thank you.
So next up we have gene conferring resistance to Geminivirus, and it's Trino Ascencio-Ibanez. Is Trino here? Oh, there you are. Thank you.
Hello, good morning, and thank you for the invitation. I really appreciate it, and I'm not sure if I'm going to live up to the hype that it was created, but I will try, certainly. And what we are displaying today, it's a gene that we found in a humble plant that was already mentioned, which is called Arabidopsis. And the point here is that we need to mobilize that to a crop in which we do not harvest knowledge, but we harvest food or commodities. So that's what we are trying to do here, if this moves. All right. So the problem that we are facing is the viruses that infect plants are spread all over the world. The particular family of viruses that I work with is called Geminiviruses, and they are DNA viruses that are transmitted by insects and that they diminish the crops wildly in many different crops, not only in tomato. I present tomato as the example today. And the impact is not only in food, but there's also commodities that are affected by these viruses. For instance, cotton, clearly, but there are others. So beans, maize, and tomato are some of them more. But there's also staple foods like cassava that are affected by these viruses all over the world. So looking where we don't supposed to look for these things, in Arabidopsis thaliana, which is a humble plant that is used like a rat lab, in this case it's a plant laboratory system, and they have a lot of varieties all over the world, or that they are called ecotypes, but I will refer them as varieties for facility of the explanation. And the point is that they have different characteristics because they've been raised in different environments. So we found one particular one that comes from Spain particularly, that is completely immune to Geminiviruses that can infect Arabidopsis. This is not to everything, it's the ones that we can infect Arabidopsis with. So we found a resistant gene against these devastating viruses. Of course, we being academics, we spend years and years trying to identify the gene, and we finally did. So we actually identify a particular gene that when we introduced it into a susceptible variety of Arabidopsis, it provides clear defense, all right? It's not an absolute immunity as we see in the original one, but it's a clear defense against the virus. It can produce. The problem with these viruses is that they don't allow the plants to produce. With this, we can get seed and we can get setting flowers, so we get production with this. Now, what is the situation that we are now? Well, we came to the Office of Intellectual Property, and we are, of course, protecting this. And the idea now is like, what do we do with it? And the next clear step is, of course, commercialization, right? But there's a, how you call it, the gully of death to still jump through. And that's where we are at this moment, okay? We want to introduce this into tomato to make the case that it actually works also in a crop that we all know and that we clearly relate to, all right? So it does work in Arabidopsis. That doesn't mean it will work, but we have high hopes that it will, because we have a lot of knowledge about the gene and how to put it in. So at this moment, we are with the patent processing already. We haven't received the patent, but it is in the process. And we have the constructs that are needed to introduce into the tomato or other crops for all means. But we are focusing on tomato for our ability and expertise to actually do transformations in tomato. So what we are looking for is a successful partnership. And we expecting or asking for support to produce this resistant tomato, right? It will be the proof of concept, not of the technology, but the actual, that it's a commercial opportunity. And then, of course, support to widen this technology. We need this. With the global warming, insects are going to be moving to places that were not before, and they're going to be infecting plants that were not infecting before, affecting areas that were not affected before. So we are really in a bind here. So the last part is, well, we hope that one day we'll have a pizza party with tomatoes that are protected, wearing jeans that I wear cotton today for that purpose. So the message is complete package, not only that. So, of course, this is not on my own. I collaborate with Dr. Linda Handleboden. We are in the plant biology and biochemistry departments. And there's plenty of people that work on this, but we only have the 5 minutes, and I think I hit the mark or not yet. All right. Okay, contacts in case you are interested. And thank you very much for your attention.
Thanks, Trino.
Great.
Mark, you look like you have something to ask.
I'm teeing myself up. So that was a really fun discussion, or fun presentation. Well done on that. I don't know much about how this virus is carried through insects. Can you talk just for like 10 seconds about what type of insects this is carried through? Is it beneficial insects? Is it pest insects? And the second question related to that, depending on your answer, would be if there were, insecticides or whatever that were used to knock down insect pressure, would that also effectively shrink your addressable market because there's fewer insects carrying the virus?
Yes, the answer is very, very simple in that regards. This it's transmitted by white flies, particularly Bemisia tavasi, is an Aleirodidae, and it's susceptible to insecticides. The issue is that the insect, through the overuse of insecticides, has already acquired resistance. And our system here is proposing reducing the need for insecticides. We want to reduce that if it's possible. In the industrialized world, like we live here, that's the way it's controlled, by insecticides. But there are many places where they are not available because the cost is too much. But there's still commercial growing of tomatoes in all those ways. But if we want to be conscious with the earth, we need to reduce the number of insecticides, the amount that we spray. We will still need them. In a perfect world, we will have very low use of them, just in critical moments. I hope that answered your question. Thank you.
Anything else from over there? I know, Spence, you've invested in a few companies on this side of the world.
It's always a long journey when you're trying to commercialize a trait, and that presents a whole bunch of different challenges. I think in this case you also want to make sure that the intellectual property is pretty as broad as it can be. So it's interesting to understand, to the extent that you know what other pathways are involved potentially that this gene or gene product is interacting with. So is there a plan around that piece?
Yes, of course. We had some money, residual money, that we are using to address all those issues. We are looking already at expression, if the expression actually affects or not. We know that it does. So higher expression induces a better resistance. We're looking for partners, if there are any partners for this particular gene. We know the gene. We have the sequences. We know what are the SNPs on the genes compared to other proteins. So we're looking to see if in some cases it's going to be an editing event or if it needs to be a transgenic event in all cases. So we are looking at that. We don't have the width to look at everything. We are zooming in two crops, cassava, because the grants that allowed us to do this is important for us, and tomato because the commercial implications of it. But there are plenty of plants that will benefit from a trait like this. And the point here is this is not the only solution to this problem. There are many other solutions to this problem, but this is one that we think it can contribute significantly, and that's what we can do. Viruses, as we all know, evolve very rapidly. So we need to have as many tools available as possible to try to reduce that. Right?
So I'm going to take a question from Jeff Rosichan from FAR. Oh.
NSF just published a Dear Colleague letter for transformation, and so you might think about applying, maybe find a partner who actually does tomato transformation to work with, and then you might be able to get your proof of concept in some other plants that will strengthen your patent position.
Thank you. Yes, we certainly will look into it. We can transform tomatoes, so we're in good position for that. Thank you for the notice.
Great. So for those who didn't hear, Jeff was talking about an upcoming NSF RFP. I'll take one over here. Did you get that?
He's asking if we have looked into tomato, if that particular gene is expressed and if it does work. That's what we are trying to actually accomplish with this request for support. We have not done the tomato transformations. We are doing the tissue culture, but we have not put it into tomato yet. The equivalents, the homologous and orthologs, they are not the same as the one that we found in Arabidopsis, so it might be a slightly different pathway. We don't know yet that questions. We still need to prove that. Yes, we can do the transient effects, but we won't know if that is able to provide resistance or not. But we can certainly do that. We can do either protoplasts or just some tissue with transient expression. We can certainly do that. Thank you for the proposition.
So I'm getting the look from Deborah, but do you have a very quick one, Frank, from Big Ideas Adventures?
Viruses change, and they mutate. How fast can you get that trait into the next? I think you're putting these into the tomato seeds, right? How fast can you change this around? I mean, just look at coronavirus, how many times it's changed. And so how fast can you be able to put these traits in there, and can you keep up with that transition?
We don't know if we will be able to do that or not. The question is about the transition of the virus. It rapidly mutates like viruses do, and if we will be able to keep up with it. That's one of the reasons why we want to put this out there, because we need it. The previous generation of resistance is already being overcome by the viruses. And this is going to be an ongoing fight. But what we can actually do is we are already starting with next generation defenses against. I'm not sure we will be able to do that. Usually in the field, traits like this can sustain resistance for 5 to 15 years, depends on what it is. We don't know how long it can. In our hands, resistance has not been broken. We really infect and infect and infect. We have not seen it. But there are not enough years or time to see that. Thank you for that comment. We'll pay attention
Thank you, Edgar.
Thank you, Trino.
Trino, I'm sorry. Apologies, Trino. Apologies, Edgar. Alper, I got that one right. Bozkurt. It's all yours.
Thank you. Good morning, everyone. I'd like to present you a very novel idea, novel way of generating data by looking at the electrophysiology of plants. We have been working on many electrophysiological assessment methods with humans and animals, especially when we try to assess the stress. For the stress of the heart, we do electrocardiogram. For the stress of the brain, we do electroencephalography. For the stress of the muscles, we do electromyography. And the question is, why aren't we applying this to plants? I'm a professor of electrical and computer engineering, but this is a very interdisciplinary team. We have materials science experts, we have artificial intelligence experts, and we have crop and soil sciences and plant experts in the team. I'd like to credit James Reynolds, who is our postdoc leading this work, and he is manning 3 booths outside on 2 other projects as well, looking at the freshwater mussel gapping by using accelerometers. You know, we have these activity monitors on our arms, and we are trying to put it on freshwater mussels. Also, we have smart moth traps. So these are other ideas I'd like to share with you in the booth. But for now, electrophysiology is based on the movement of the ions or conductive particles inside the tissue, and has been a great tool for the animal and human biomedicine. It has been a tool as well in the plant science, but it hasn't been as translated or scaled up because of the issues with the instrumentation. And our novelty that we are trying to bring to the table is more about how we can make electrophysiology on plants practical. We start with novel surface electrodes. We have nanocellulose substrates on which we have graphene as conductive materials. I will show it in my next slide. We have really small biopotential amplifiers, tools like electrochemical impedance spectroscopy to look at the conductance of the ions inside the tissue. We have now very ubiquitous wireless transfer methods from Bluetooth to Wi-Fi to LoRa to LTE. And now we have also capability to put together various multimodal sensors next to our sensor to do environmental sensing, to do vision, to look at the stem diameter. So these are other sensors we are developing within this project. But I just would like to focus on the electrophysiology itself because it's very cool and it's very emerging. The common way, especially the interfacing of the plants with electrodes to get these tiny voltage signals from the plants is either you use needle electrodes, top left. Those are things that you insert the plants to look at the stress, and it has a potential to create stress. There are, the middle top shows some patch clamp electrodes, really nice scientific tools, but not easily usable in the field. The top right is, I think, top left, sorry, is the very cool idea of inserting wires into the head of aphids and letting aphids to bite the plant and doing a measurement through the aphid. Cool scientific methods, but we try to, in the bottom, you can see our nanocellulose substrate, very thin membrane. It doesn't need any sticker. You just wet it, put it on the plant. Cellulose is not a different material, not a foreign material for plants, and it beautifully sticks, and we put conformable graphene on top of that, which is a conductive material as electrodes. This is basically an ECG, EKG, electrocardiogram electrode for the plant. And the instrumentation is, when we first started more than 10 years ago, is what you see on the top left. The benchtop instruments, it costs more than $1,000, and it's really difficult to bring it to the field. And on right, you can see a small board that we designed, matchbox size, solar powered, costs only a few dollars, and we can do crazy stuff: biopotential amplification, impedance analysis, wireless transmission, onboard analysis, but mostly transferring things to the cloud. Some proof of concept experiments. We started with Arabidopsis. We are engineers. When we started our experiments, we were like, are we playing? If we do experiments on crabs, is it considered playing with your food? Arabidopsis was the most accessible thing to us. We cut a leaf, and in the other leaf, we see the top left signal. It's what we call a hey signal. you cut the plant and you hear a hey in the other leaf. We drop cold water to the cassava, this is top right, and one leaf we drop a cold water droplet, and the next leaf says hey. And a nice thing about Arabidopsis is you can genetically play with it, and the bottom is an Arabidopsis mutant that is ozone addict. It is very sensitive to ozone. So when we apply ozone to this ozone addict plant, it goes like, hey, and you can see here on the blue signal. And plants love also light, and we applied light to see whether it's a general response, but it wasn't as strong as the ozone. So these are just proof of concept. What we record with biopotentials, like tiny voltages on the surface of the plant, are modulated with these environmental exposures. And we also do a deeper dive, being electrical engineers and like playing with signals, into the impedance, general conductivity within the tissue. Like these are all sort of crops or vegetables or what we call lower hanging fruits for us to run experiments: bananas, sugar canes, soybeans, maize. We put electrodes and we just look at how the electrical conductivity is, and these are day and night cycles, diurnal patterns, and we see things change in the plant with environmental just day and night changes. So the things that we are asking from industry is we are running a lot of low TRL, technology readiness level experiments with our electrode structures, how they interface with the plant, whether it allows perspiration, whether it allows oxygen transfer, whether it allows light transfer. But in addition to that, we do a lot of high TRL experiments where we expose the plants to different stressors, but this is very application specific, and we are looking for partners, commercial partners who has research departments, who has, who intentionally stress out the plants. And we can get into those platforms and record our signals with electrophysical sensors. We are interested in, definitely there are a lot of translational barriers to go from pythatron to greenhouse to the field. And we know industry knows how to play that game, and we are looking for that partnership. And we are also looking for extra financial support to scale up our experiments. We want to generate not only 5, 10, but thousands of these electrodes, thousands of these boards, and put it around the different fields and do signal processing. The signal processing, Edgar, who had the microscope robotics presentation, he is a genius on AI and machine learning, artificial intelligence. This is one of the things that we are working on, generating a large data set and apply artificial intelligence to that, and train artificial intelligence based on contextual information, because we also record all these different environmental signals, and then try to use this maybe as a method to predict the plant stress. Thank you.
Thanks, Alper. We have a moment for a couple of quick questions.
Quick question for me. Really cool science. Love what you're doing. It's super interesting. I think the challenge for me, and this did not come across in the presentation, is how you are translating a specific stressor fingerprint, or signature, to what the stressor is. Because it feels like there's a lot of noise you could get in this, like sort of short-term stressors that don't really have any impact on the plant vigor or health. So where are you in being able to decipher this stress causes this signature graph?
Yep. So the stress detection is actually, my other hat is I'm deputy director for a National Science Foundation funded research center, assist center here. We work on wearable electronics for humans, and one aspect we try to do is using wearables for mental health. Stress detection is a very big problem. And there are a lot of things that stresses us, and sometimes they are not even stress, they are just arousals and other psychological factors. The thing here is, what can we learn in a closed loop? Like if you have the artificial intelligence look at your data. And one particular problem that we are trying to solve with maize is whether we can optimize the water delivery and fertilizer delivery such that we are not going to, we will improve the yield, we are not going to stress out the plants, and we are not going to get them dry up, we are not going to overfeed them. So what is the optimum amount of the nutrients we can provide the plant? So it's really, this is a very general presentation, but we have grants for a particular problem like watering and nutrient deliverance to maize, and we collect data on that, and we try to fine-tune our artificial intelligence and machine learning based on what the application is.
Got it. Thanks.
So that's all we have time for, Alper.
We will be around. We are on the booths.
Yeah.
We are not disappearing. Thank you.
Thank you, Alper. So delighted we're moving out of the plant sciences for a moment into animal science, and we have Stephanie Ward with milk as our diagnostic tool. Over to you, Stephanie.
Sure. Thank you. Well, good morning, and as Adrian mentioned, I'm not a plant scientist, but do represent the cheese on Trino's pizza. Hopefully we will keep that there. Just show of hands who likes cheese. Ice cream? Perfect. I like to know my audience. And just so you know, we do make the best ice cream in the world here. If you don't know that, you should try it before you leave. We'll get you some. All right, so for those of you who may not know the importance of dairy in our world, I want to take a minute, 15 seconds, Deborah, to talk about that. So while I'm not a plant scientist, dairy cattle and hog, poultry, other food animals are the end user of the majority of the plants that we work with here, right? So just to put in perspective for you, the 45,000 dairy cows that we have in North Carolina represent 134,000 tons per year of corn and corn byproduct utilized, and then 71,000 tons of cotton and soy products and byproducts. And the really neat thing about dairy cows is that they use that to make protein. And in a global crisis centered around hunger, they're a huge part of that solution because that protein is exportable. We can get dried milk powder and we can get aseptic milk to parts of the world that need that protein. And that comes in 585,000 tons per year of milk, 17,000 tons per year of dried powder protein. And at the end of her productive life, the dairy cow actually gives us just under 5,000 tons a year of beef. Now, it's not usually the prime rib or the ribeye that you sit down to at the Angus barn, but it is a really good hamburger. So we have used milk in the industry. Oh, I'm going to just pause that because I'm going to out-talk this timer here. As a diagnostic tool for a number of years, we do recognize that it can be utilized to diagnose pregnancy and things like mastitis, which is one of the most costly diseases to the industry itself. But we also use it to measure production efficiency, and now we believe we can use it to also detect metabolic disorders and diseases, and then utilize that data and information that comes to us to turn around and make management decisions on the farm. So we do that through categorizing the fatty acids in the milk fat. And this is work that we've done here at NC State, but also in collaboration with Dr. Dave Barbano at Cornell University, where we've categorized fatty acids according to chain length into de novo fatty acids, which are typically short-chain fatty acids that are made by the animal, so they're new. We have preformed fatty acids, which are typically either from a dietary source or also from mobilization of adipose tissue in an energy deficit situation, and then mixed origin fatty acids, which come from both places, right? And so by understanding the concentrations of those different fatty acids that make up milk fat, we know what the metabolic status of that animal is. So this is some work that we've done here recently where we looked at cows that, going back to mastitis, cows that were infected with mastitis versus a healthy cow. And these are partnered cows, and what we found is about 3 weeks prior to diagnosis of mastitis in this herd, we were able to see a deviation in de novo milk fatty acids. So what does that let us do? It lets us treat her differently faster. So antibiotic usage is one of the things in the animal agriculture industry that we get a lot of negative publicity for. If we find out 3 weeks before she becomes clinical that she is heading into sickness, we can treat that differently, and we can then be a little bit more prudent in how we utilize antibiotics in our food animals. So we're using mid-infrared FTIR spectrometers to detect these different components of milk. And in all honesty, this started out as a way to get dairy farmers better pay, a more accurate way to detect components in the milk. But our challenges are this. Right now, the best we can do is herd level data, and it also requires a person present to take those samples and analyze in the lab. So we need to take this to the cow level so that we can start to analyze and understand what's happening in our individual cows that operate as part of a larger herd. The second thing we have to do is make it a little bit easier to collect this data and a little bit more automated. The dairy industry is a leader in terms of automation and robotics where animal agriculture is concerned. How many of you liked ice cream? How many of you wanted to get up at 4 a.m. and milk cows? Beth would. I know Beth would. Okay, there's not a lot of us left that want to do that, right? So what have we done? We've moved into automation and robotics, and these are two examples of different robotic parlors. So the technology exists. We have to learn to integrate into that and get the most amount of data that we can from that. The last challenge that we face is farmers don't want data, they want information. They don't want numbers, they don't want to have to open a spreadsheet. They want you to just tell them, or they want the computer or their phone to tell them what the next step is. So while we have to work on the technology piece and developing that further, we also have to work on the data analytics that are behind that. We have some data here that's sort of proof of concept between us and the Miner Institute in Chazy, New York as well, where we've looked at different diseases and we know we can utilize this data to predict that onset faster. And with that, that's it. I'll have...
Questions for Stephanie?
Oh, I have one. And comments on top of it. So, by the way, University of Wisconsin-Madison is an investor in our fund, and I'm going to take your comments about the ice cream in the creamery here back to there.
Sure.
We'll see how that goes.
Do it.
But yeah. There might be an ice creamery off at some point here.
We actually do that every year.
Do you?
At our American Dairy Science Association meetings, and we did win. So.
We have data.
And Wisconsin was there.
There you go. As long as they're represented, you know. Wow. They've fallen off the perch. Well, the just in terms of mastitis, I think you're on the right track here about the things you need. It's super, super hard. In animal health, there's, at least from my experience, there's probably not another area that has as many dead bodies on the side of the road.
True.
As detection of mastitis, and even more so than sort of like feed metrics and stuff like that with beef cattle. So I think definitely your hardest problem will be how is this fully automated at the single cow level that still is at a very low cost point, because despite much better margins in dairy than lots of other things, and that's why they have so much technology. The price that they pay for mastitis sort of remedies, is extremely, extremely low. So cost and full automation, I think, are the two things, and I just flagged that.
Sure.
Anyone else from our sharks or Kelly?
I think this is fascinating, and it's wonderful we've made it to the point to bring the data together to be able, because I think if you work with cows, but to be able to actually measure it. My thought is, are you also having discussions with people in human health, not necessarily for just milk, but for the detection of fatty acid changes like this somehow or other in the body, or metabolic changes that might diagnose problems earlier than what we can currently do.
So human health applications.
Yep. So we did. We have had some conversations with some companies who are developing implantable chips with sensors, particularly on the side of diabetes in humans, because loosely cows are typically always diabetic, right? There's more science than we have time to explain behind that, but they are a good model for trying to track particularly things like ketosis and fatty acid movement. So we have had those discussions of there's benefit in using cows as a model for that purpose, but then we also would be able to gather data from that to use, in understanding the metabolic status a little bit better. Yep. Other questions?
We have one over there.
Yep.
Have you tried to use to modify the formulations? You know that the animals are more in negative energy balance and all that. Maybe you're gonna have more, gonna be luckier selling the technology to be more efficient doing simulations that just we're talking mastitis might not be a very smart.
Yeah, so so the reason I chose mastitis to talk about today is related to the antibiotics issue. And we also are just getting ready to go under a new directive that limits utilization of those even more to farmers. On the feed side, though, that's where some of this technology is really impressive, to the point that we can see changes that happen in labor. For example, we can see a change in de novo fatty acids when someone new is running the silage wagon that day, or when there was more rain in the area on uncovered feed, right? So all of those things contribute to that change. And so as a result of that, the majority of the large milk buyers and cooperatives in the U.S. have started producing this data, but it, again, is on bulk tank. So it's on the whole herd rather than individual animal. But that data is now available to farmers, their nutritionists, and their other advisors can utilize that information for exactly what you're saying, to address and chase down issues in the ration or other places on the farm.
Can you go one step even earlier? So I know you're looking at fatty acids and then it detects. What causes that though? And so can you go back one that you don't even have to get into the antibiotic issue? You see where I'm going at from?
You mean looking before the fat deviates?
Yeah.
Yeah, and actually I've heard some presentations here this morning. I'm going to be sending some emails to see if we can't adapt some of those things to milk as well. So I think we could. I think there's some places we could look that might be a little bit easier to integrate into our milking systems, because not everybody's going to move to a robot, especially some of the bacteria detection options. It's a lot of different things, so a lot of different bacteria. There's also some management issues. Usually it's a cleanliness issue, but the most common ones that we deal with as far as pathogens go are staph and strep bacteria.
Thank you, Stephanie.
Okay, sure.
Appreciate
Don't forget
it.
to get the ice cream.
We're talking to Kieran later on, Spence. Okay, so now we're going to move on to, that was our 5 pitches. Now move on to something slightly different with Qingshan Wei. Qingshan, please come up. And Qingshan is doing some amazing things exactly 2 floors above us in terms of sensor development. But I think you're going to talk more generally about some of the challenges, right? And we'll thank you guys later. Please stay where you are, because you may have some more questions, and we'll bring you into the conversation for the other presentations. Qingshan Wei, thanks.
All right, good morning, everyone. First of all, I would like to thank the organizer for the kind invitation. It's my great pleasure to be here. And then in the next few minutes, I'm going to quickly talk about our experience of technology development and also commercialization effort. My background is in engineering chemistry, but I'm very fortunate to work with a few plant people, expert plant pathology, to work on some plant sensor projects. So in general speaking, plant sensor is still very small area in the sensor field. If you look at the publication number, it is increasing rapidly over the years. I think that is the same trend if you look at the commercial products. One trend we observe is the digitalization and the miniaturization, which means new technology try to move lab-based technology into a small sensor that can be used in the field for continuous and real-time monitoring. And we put in a few efforts, try to follow that trend. One thing we did is try to move the molecular diagnostic from the lab setting to the fields. We demonstrate a minimally invasive microneedle patch that can quite quickly isolate plant DNA and RNA without using bulk instrument in the field within less than minutes. So basically, it involves very simple 2 steps. You press down the patch on the plant tissues, and we demonstrate the patch can be applied to different plant tissues, including soft and hard leaves and also stems. And after the sampling, we can put the sample into a portable diagnostic platform. Could be a smartphone-supported nucleic acid amplification platform. We can rapidly amplify the targets to inform the plant infections, whether it's a healthy or infected. The second technology we are very interested in is always a completely non-invasive sensing methods. For that, we go after the VOC detection, the volatile organic compounds. We work on two different sensing platforms. One is, again, smartphone-based VOC sensors that can perform point measurement using the colorimetric signal change. We can profile the VOC emission from plant tissues to understand whether we have a disease or not. The second platform is more a variable concept, which means you really deploy the sensor on the host plant in situ, so we can monitor the plant response in real time. We already demonstrate a few generations. The first generation, we only be able to measure the VOC signals, but recently we are able to integrate other sensor components such as temperature, humidity sensor onto the same small patch, and then you can wear the patch on the live plant for continuous monitoring. And for more technical details, you can welcome to visit our exhibiting booth. I think it's number 7. It's outside there. Like many other engineering groups, we are also interested in tech transfer. We made some effort in this domain, which means we participate the ICOR program at NC State in 2002. So the team included 2 students from my group, Rajesh and Dak, and we complete totally 15 interviews to different sectors, including different growers, technical companies, research labs, and even government agency focused on the regulations. Our initial target market will be single users or small growers because we claim our sensor technology is portable, low cost, affordable, right? During the interview, we found out even for small grower, for example, like a family-owned farms, the size of farm really varies. Could be from 1 acre to several thousand acres. So that represents different level of willingness to adapt the new technology. There are different challenges. Another major finding from the interview we found is there potentially a new market or new customer segment, which is the controlled agriculture, which we didn't consider before. Controlled agriculture basically is you grow crops, plants in a greenhouse settings. This is a rapidly growing market. About 30 years ago, only 0.5% of tomato are coming from the greenhouse. But if you look at the market data nowadays, almost three-quarters, 75% of tomato are coming from the greenhouse. In the greenhouse environment, basically people focus on automated irrigation, climate control. Sensor is a very important, essential component to achieve those goals. In the closed environment, the disease or pathogen spread is also a big concern. So again, we need some sensor for rapid health monitoring or disease diagnostics. So with those factors in consideration, really after the program, that significant changes our business model. Before the ICOR program, we think small growers, regulators, because the sensors are low cost and portable, maybe that's a target we should go after. But after the program, I think immediate application for those sensor technology, the controlled agriculture, maybe that's the best, the first step, because it has the needs, it also has a market size. And also the program allows us to narrow down the numbers. For example, we can do much better estimation. We can, for example, reduce at least a 5% crop cost if we can apply the sensor technology. We can save 250,000 capital dollars in terms the savings if applying the sensor technology. Finally, the lesson we learned through the journey and the process, we feel it's very essential to talk to the direct users when we try to develop any technology or discoveries. The more you know, the better technology you're able to develop can fit the needs of the users. That's also the same thing, the true. You want to establish a collaboration with industry partner as early as possible, because the industry partners always stay at the frontier of the application. And the lab, we are just focused on fundamentals, right? So the second thing we learn is for all the technology, once you want to transfer from the lab to the real use, a point, proof or field trial is very important. But the field trial in the meantime is very challenging. We are participating in a field trial currently led by a plant pathology, Jean Ristano, also in the audience from Faculty of College of Agriculture. And we see many different things, issues we didn't expect. And it's a great opportunity for us to solve those things before we push the sensor to the market. One of the most important things we learned from those field trials is the robustness and the reliability of the sensor sometimes outweighs their performance. So I think that the user will maybe more prefer a sensor that can last longer, can get the job done, but you can sacrifice some of the performance or functionalities. That's the thing we learned. So quickly sum up, we have a sensor exhibit booth outside. And if you have more questions on the technical detail, feel free to stop by. And I would like to thank all the funding agencies to support the research work here. Thank you, everyone.
Thanks, Qingshan. We have time for a couple of questions. I just want to say thank you for mentioning the Controlled Environment Ag. Actually, at the last ERS, we actually launched a consortium around Controlled Environment Ag research, and we hope we can become a really recognized center of expertise in that area and really support the industry as it evolves over the next few years. We do have time for a couple of questions if anyone wants to ask about plant sensors or has any thoughts. Thank you.
Yeah, you mentioned about the DNA extraction using the micropatch, the needle stuff. Does it work for all kinds of plants, meaning plants rich in phenolics? Because usually when you have the DNA extraction, phenolics does interfere with the DNA extraction and even in the regular process of the kit, but it is just a patch here. So how does this account for plants with phenolics and other compounds that interfere with the DNAs?
Good question. So I think the question about the interference and then potential inhibition, right? Yes, so currently our patch, they extract everything. It's not selective, which means they extract DNA, RNAs, and then they extract maybe something on the surface of the tissues. But we do have a control experiment. They do get a lot of material inside the tissue. That's the thing we want to sample. For future, we try to modify the patch property a little bit more so that they can be more selective. For example, they only extract the nucleic acids or only extract the proteins or other things. Yeah, that's the future ones.
One more question? Please.
Just real quick, are you looking for any donations of technology, not money? Because I've got some technology that is abiotic stress that I would be more than willing to let you have and try for your sensor experiments.
Sure, we are looking for all sorts of different collaborations, is either the fundings or technical support or support our field trials. Anything can further enhance the performance of the sensor platform will be very interesting. So I'm happy to talk. Yeah.
That's good to hear. Thank you. All right, with that, thanks, Qingshan.
Thank you.
This one. So next we have Brian Eller. Brian, please come up. So Brian is Assistant Director of Licensing with our Office of Research Commercialization. So thanks, Brian, for joining us.
Well, hey, everyone. It's a pleasure to be here today. I'm actually filling in for Wade Fulgham, who's the Assistant Vice Chancellor and the head of our office. So as many of you may know, the Office of Research Commercialization manages all the intellectual property developed here on campus. So that means from the disclosure stage all the way through marketing it, evaluating it, protecting it via patent or other intellectual property means, executing licensing deals, being part of helping sponsored research agreements, putting them into place. So I know there are a few folks from my office in the audience here today, so I hope that maybe you could just stand up and maybe just briefly introduce yourself.
Good morning. I'm Devish Srivastava. I'm also a senior licensing associate, and I'm in the life sciences group, and I've got a few of the faculty presentations being made today in my portfolio. Happy to be here.
Thanks. So we're very proud of the work that we do at our office in collaboration with all the researchers here at NC State. We consistently rank highly among the metrics that matter. We're especially we compare ourselves to universities without medical schools, but you'll see we're consistently within the top 10, and that's just a credit to the quality of research coming out of the university, but also a lot of our research administration, our ability to move at the speed of business, to negotiate contracts quickly and efficiently. And so we're really proud of these numbers. Throughout the lifespan of our office, we've launched nearly 200 startup companies. You'll see, that's over 600 products to market, nearly 10,000 jobs created, which is really impressive. So I just want to give it a little history lesson of why did our office come into existence, and how does research commercialization come into existence at universities? And we at least trace our roots back to the Bayh-Dole Act. And so on the screen here, you'll see Birch Bayh and Bob Dole. And they're famous for, I guess they both ran for president, but they're also famous in our world for the Bayh-Dole Act, which was passed in 1980. And what the Bayh-Dole Act allows, it allows for universities to take ownership of inventions made by research funding from the federal government. So it used to be the case when the federal government would fund research at universities, the federal government would own those inventions, and then they would try to commercialize them. But they realized by giving a profit incentive and motivation to universities, where the research was being done, if they gave them the ability to take ownership of those inventions, that they could then, further commercialize them, incubate them, promote them. And so, in the 1980s, this was kind of during difficult economic times, but they thought this would also, kind of revitalize the American economy. And so there's a quid pro quo in the Bayh-Dole Act, and it requires universities and our office to, actively promote and attempt to commercialize the inventions that we take ownership over. And we give preferences to U.S. businesses and small businesses. And we also provide incentives to inventors to encourage them to innovate and to commercialize their inventions. So part of the revenues that we receive from licensing, and we return to our inventors, whether they be students or faculty. And then we use the remaining income for research and education to perpetuate further commercialization efforts at the university. And then, one caveat that the government has is they have these things called march-in rights. In case there's a pandemic or other kind of national emergency, or there's a requirement that, that they feel that there's an emergency situation where those patent rights, which are next kind of exclusionary, we need to get those widely disseminated, the government can march in. And so this has been, a widely successful, piece of legislation for universities and to thrive and commercialize their research. In recent times, even just this year in 2022, in the CHIPS Act, which is mostly famous for semiconductor chips and that sort of thing, but buried inside the legislation, there's actually direct funding for tech transfer offices. And so we've kind of considered Bayh-Dole an unfunded mandate. And so our office to date, we pay for all the salaries and we pay for all the patenting that occurs at our office through the revenue that we generate from licensing, which is not a huge amount. We're not trying to, I guess, there's a balance between trying to commercialize something and trying to squeeze every last dollar out of something. And so, we're out there, our mission is to really just get products to market. But we're pleased to see that there's, this recognition that, these kind of services could, use further resources. And so this allows for $1 million grants for up to 3 years in duration. And there's $10 billion allocated to regional economic growth and development hubs to try to commercialize research. So this is a crowded slide, but I was asked to talk about kind of what is the commercialization process at NC State. And so I won't go into this exact steps, but I would just say that, we have many professionals on our team that were involved in the evaluation, the disclosure, the marketing, the protection, and the licensing of intellectual property here at NC State. And that is sometimes it can be an expensive endeavor. And I know there's some, I think there's some patent folks in the audience. I saw some of them, the sponsors or business firms, but we have to use outside counsel to file and protect those patents. And so, since we're kind of a self-funded office, at least for now, we don't file patents just to get plaques on people's walls or to pad their resumes. We're here to really try to commercialize things. And so, we can't patent everything that comes through our office. So we receive about 250 to 300 invention disclosures every year. And of that, we patent less than 50% of that. And then maybe of that less than 50%, maybe half of that will, make it into a further, we'll do initial patent filing, but then those half of those will be converted or further supported along the way. And so during that time period, we have to find commercialization partners. We either have to help create a startup company, or we have to find an existing commercial partner that's willing to take that on and help commercialize that inside. And so, a lot of university research is early stage, so we have to have programs and means and ways to help, bridge that valley of death and, get those technologies out of the university. And so we have developed a variety of programs to help try to do that at our office. So one we're very proud of is the Chancellor's Innovation Fund. And this was actually supported through initially was seeded with money from the sale of Cree stock. So Cree, or now Wolfspeed, was actually came out of NC State in the late 80s. Silicon carbide technology out of the Material Science and Engineering Department. And so the university took equity in that startup company, and then through that sale of equity, we were able to fund and seed the Chancellor's Innovation Fund. And so you can see how, past successes can, snowball into future successes, and we're hoping that today's technologies and things you hear today will help further snowball into even further successes. And so these, the Chancellor's Innovation Fund is basically trying to take early stage research that may not be ready for commercialization, and maybe bring it over the finish line. And so what it relies upon is, researchers getting feedback from industry and saying, hey, maybe there's some critical experiment, there's some critical field testing data we need, but we can't get that money from the federal government. They're only sponsoring, basic research. How do we find ways to get that final, piece done? And so this is a competitive process. I think some people in the audience may, have been involved with that process, but I think it's more of akin to like a mini Shark Tank, if you've ever seen that TV show. But the final round is really a pitch to say, what is the ROI on this invention for the university? And so we award 6 $50,000 awards every year, and through that we've generated, countless startup companies, further licensing revenue, and we've also made incredible social impact as well. So we're really proud of this program, and if you know researchers or you're interested in applying, we encourage you to do so. Another program we have is the NSF I-Corps program, and I know if you were just listening to Qingshan's previous presentation, he was involved with that and involved getting customer interviews. And I'm just going to maybe hand it over to Tim, since he's been heavily involved with that program. I don't know if you want to come up here, Tim, and give a little talk about I-Corps.
Yeah, we've heard comments from investors about identifying who that target customer is and identifying what the value proposition is. We go through that process with the researchers through I-Corps. So many of them, this is the first time they've done anything business related, been researchers their entire career, and we ask them to go actually talk to the customer and learn what are their problems, what are the things they actually care about, and see if that matches. And if it doesn't, we make a decision, either change the market, figure out a different customer, or change the product. And so through that process, we're hopefully informing either future research opportunities based on those conversations, or potential collaborations with those companies and partners as they go through. But we hold 3 cohorts a year. We're part of the National I-Corps Hub program, which was launched 2 years ago now, out of the University of Maryland. So we have extensive resources both on campus as well as regionally to help our startups and our researchers really learn enough to be dangerous, essentially, to go out there and get the initial funding to then bring in external management, external CEOs to help grow those companies. So we're really proud of the program and hope to see many of the faculty that have presented today participate if they haven't already, and happy to see some of the successes from some of the other faculty that have already been through it. So thank you.
Yeah, so like Tim said, we host this I-Corps site, but there's also regional and national I-Corps sites, and the national program has like a $50,000 award associated with it, and involves getting 100 customer interviews. So yeah, we've noticed that this has been really, I think, eye-opening for everyone who's participated, and really we've launched several startups have formed out of this program, and so it's a really valuable experience, and we encourage not just faculty, but if students that are interested in commercialization, this is open to the wider community as a whole. So it's not just just faculty. We want everyone to be involved here. I think one other organization we're heavily involved with, and actually I see Lisa Chang in the audience, who's involved with the Wolfpack Investor Network. But we also noticed that hey, it's just not enough to our office is really good at negotiating agreements and creating startup companies, but once we launch them, can they succeed out there in the wild? And so getting that initial funding can be challenging. There's small business SBIR programs, so small business awards that you can get from the government. There's venture capital funding that you can obviously go after, but what are other ways we can get that initial funding in? And so it was observed that there's a lot of alumni that are accredited investors at the university. And so we formed the Wolfpack Investor Network, which we have a great acronym of WIN, and that uses, if you're an accredited investor, you can join this program, and basically we send deals associated with NC State through that program. And so either those companies have licensed NC State intellectual property, or they have an NC State alumni, or current faculty, or current employee, current student, and they can pitch to this investor network. And so the investors basically get to choose which deals they want to fund, but pretty much every single company that has presented in front of WIN has gotten some form of investment, and there have already been several successful exits. So if you're interested in going to WIN and presenting, or being an accredited investor, I'd encourage you to reach out to Lisa here, who's in the audience, or reach out to my office. But we found this to be a really successful program, and we're hoping this also helps launch the future companies of tomorrow. So that was a really quick overview of my office, but I'm happy to answer any questions you might have.
Thanks, Brian.
Sure.
What's the legal structure of the Innovation and Commercialization Office?
Right. So we're employees of the university. So we're not a research foundation or anything like that.
Okay. So it's not a separate sort of legal entity.
Right. So I guess if you think of org chart wise, so there's the chancellor, and then there's this Office of Research Innovation, and so underneath that is the Office of Research Commercialization, and then there's sponsored programs. So that's research compliance and getting sponsored programs in there, and there's other proposal development units and other kind of research administration units in that organization.
Okay. Thanks.
But also in our office, we have 5 attorneys.
Okay.
Yeah. So we have people with attorneys. We have people paralegals. We do rely on outside counsel to do the patent filings themselves, but we also work a lot with the Office of General Counsel and other units on campus.
And we're former engineers and scientists too. So.
Yep.
Frank.
Your NC State is a land grant university. How are you guys getting equity into companies that are not allowed? How did you guys do that?
So I guess that was news to me. My understanding is that we can take equity in the deals.
Research foundations can, and you're not going through your research.
That's not necessarily true. A number of public universities do. I won't get into the intricacies of it, but no, it's not limited.
Yeah, you can talk to the Office of General Counsel, and we have stock certificate. We hold stock. Typically in our deals, so this gets maybe into the nitty-gritty details of license agreements. We'll also take, ghost equity, so success fee-based licensing. So again, if if the company sells, we get X minor percentage. If the company were to sell, change of control. But we also do have historically taken equity. So.
Any more questions for Brian? Okay, well, thanks, Brian.
Sure.
Appreciate
Happy
it.
to be here.
All right. So now we have Dinah from Carolina. Dinah Schuster, our head of advancement. No, you can use this if you want to.
I kind of wanted that, but hey, hi everybody. Great to see so many familiar faces. Thanks for being here today. I'd like to start first of all. I'm one of the lead fundraisers for the Plant Sciences Initiative, Dinah Schuster. I also oversee the Research Foundation Board. I'm joined by Aaron Meyer in the back, who's on my team. Those of us that have fundraising responsibilities, first and foremost, who's the guy that said you want to donate equipment? Hey, we're the ones. We'll find you. But anyway, I just want to say that was magical words, and every fundraiser in the room sat up very straight when you did that, so thank you. So really, I'm here just to say that there are a lot of ways that you can support the work that we're doing in CALS at the university with Plant Sciences Initiative, and really one of those is through philanthropic gifts. And what's really, really exciting as I look around the room, there are a number of you all that we are talking to currently about gifts to support the work that we're doing. So delighted that you're here advocating for the work, and also that we are talking about how you can do things to help us reach our goals as well as yours. So what I will just say is if you see me or Aaron, have a chat with us. I'm going to tell you the thing that we need first and foremost that I hear from how many faculty are here? Can I just see a hand? So every single one of you know what I hear is we need more graduate student support. That is what I hear every time I run into somebody. So what I will tell you is that is the thing, probably first and foremost, that we're going to ask. You're hearing about all these great initiatives and these projects. And I loved all the presenters, but we happen to know that there's an army of students and postdocs and graduate students behind them that are doing a lot of the work. So I hope that we can continue some of these great conversations. Again, I'm thrilled to see friends sitting here at the table and a lot of representation from lots of different industries. So enjoy your day, and we'll bump into each other. And again, don't leave. We'll find you.
You're tagged. Thanks, Dinah. Okay, so just leaves me to wrap up and say a big thank you. I know some of you are staying on for the VIP Investor Forum, which will be in this room in half an hour or so, but we'll give you a little bit of a break. But for everybody, a round of applause, first of all, for our sharks. Thank you to Mark, Spencer, and Heather. Thank you to all our pitchers. Thank you to Qingshan. Thank you to Brian. Thank you to Dinah. Did I miss somebody? I'm not sure I did. So thank you to them as well. Please, two things. One is our next emerging research showcase will be in February. It's up there in the corner, February 21st, around permanent and semi-permanent carbon capture. So another kind of relevant topic for today. And also, please use the next time to go visit some of our booths. We have, 17, I think? 16 booths, including one that's on the sidewalk out there. So don't miss the cow shade. But most of them are up in the lobby area. So please just kind of check them out, and please, have a word with the researchers there. They're all really excited to be showing off to you what they're working on. But with that, I'm going to close this down with a last thank you to Deborah and to Chris. We've mentioned him a few times. He would have loved to have been here, but we'll pass on the fact that we felt this was a really really good event with a lot of audience participation, which we really appreciate. So thanks very much.